Okay, so hello everyone. Welcome back to this coronavirus lockdown interview series that we're doing here on the channel. I'm joined today by another researcher, which I'm really looking forward to getting into this topic. I'm joined today by Jana Obajdin, who's a PhD student in cancer immunotherapy at King's College London. Hi Jana, thanks for joining us.
Hi, thank you for having me.
So I think just to jump straight in, do you want to tell us a bit about your kind of journey, who you are, how have you come to be where you are? We'll go from there.
Yeah. So originally, when I was applying for universities, I actually did not intend to go into research. I was applying for dentistry.
Okay.
Um, and so I wasn't aware of the sort of lower acceptance rates of international dentistry students in the
Hmm.
UK. So I didn't get accepted anywhere. And I ended up doing biochemistry at the University of Leeds, which to this day I'm very thankful for doing because I think it opened up a whole new range of career opportunities and directions that I can take, which I wouldn't have known had I chosen to do dentistry. And throughout my undergrad degree, between my second and third year, I also did a year in industry. And I did this in a sort of, I guess you'd call it a medium-sized pharmaceutical company in Belgium. It's called UCB Biopharma.
Okay.
And what I was doing there was I was working in the Department of Investigative Toxicology, and we were looking at uncovering and validating biomarkers of drug-induced organ injury. So I was specifically looking at drug-induced kidney injury, and we had the sort of panel of potential biomarkers, and we wanted to validate them. And before I did my year in industry, they had already come up with a sort of set of biomarkers, and one of them was looking quite promising. So I spent the year validating this biomarker, and we really did show that it does get sort of upon kidney injury, we see that this biomarker gets excreted, and we think it's quite a promising new biomarker of kidney injury.
So just for the boys and girls at home, the
Yeah.
medical plebs like me, what do you mean by a biomarker in context?
Yeah, a biomarker is something sort of like a protein or even the sort of increased expression of a gene, but it's usually something like a protein that we can measure, whether that's in the blood or in other sort of bodily fluids like the urine, which we can distinctly measure, and it can tell us what's sort of happening within the body. So we were looking at how drugs can affect the kidney. So we found that in at least in the rat models that we were using, that this protein gets excreted into the urine of these rats when they're treated with these drugs that we know cause kidney injury.
Hmm.
It was quite specific to upon treatment, and we didn't see this in the healthy controls. So we validated this novel biomarker, and I think what was most exciting for me in terms of my career in research was that led to my first author publication, which was quite exciting considering I was still an undergrad
Yeah,
student. And
that's very unusual.
yeah, it was quite unusual at the time, and I'm forever grateful for that opportunity. Um, I then went on to do my undergraduate dissertation project when I got back from that, and I took quite a drastic turn here. So I moved into looking at more structural biology,
Hmm.
which I know might be a bit abstract for medics
But
potentially,
please
but
go just explain your terms, and I'm sure we'll be fine.
yeah. So I was looking at the lab that I was in was kind of looking at these structures of proteins and how enzymes work, and but more my project was more specific in terms of we were looking at these bacterial systems where they make their antibiotic production systems, and so antibiotics are these highly complex, intricate molecules that I think organic chemists just can't synthesize in the lab. I think it'd be almost impossible to. And so most of them come from bacteria producing them themselves.
Yep.
But because we have this lack of antibiotics, well, we've reached the peak of uncovering new antibiotics, and we're now moving into this whole era of antibiotic resistance, which is quite a scary concept, I think.
Hmm.
We're looking more into looking at how bacteria produce them. So they have these intricate systems and how they actually produce the antibiotic. And we're trying to mix and match different bits of different bacterial systems and use them as tools and kind of
Hmm.
put them together to make a new system that can potentially make a new antibiotic. So what my project was doing was looking at in one bacterial system, which generated this antibiotic indenomycin, which never made it to FDA. But what was interesting is that we found this enzyme which created this interesting chemistry, which you don't really see in a few other enzymes. And we thought if we can incorporate this into other systems, we can make a completely new antibiotic, which would, kind of be the end goal. So my project was more using techniques like X-ray crystallization and nuclear magnetic resonance, and finding really the actual physical structure of the enzyme, and then which again I think I was quite lucky with, because we managed to find the structure of that enzyme by the end of the project, and I think that was again very exciting. So
so was it just so very, very quickly, so I can paraphrase again, just so I understand.
Yeah, sorry.
So I've got to spell this out for all the medics and pre-meds at home. So you were looking at, as you identified, antibiotic resistance, huge problem, very difficult to synthesize, discovered de novo kind of solutions. So you can take various parts of these bacterial systems that often, for their own reasons, will synthesize their own antibiotics. And you can kind of take bits of protein and bits of enzyme and genetic machinery and things to kind of glue it all together.
These are these kind of multi-enzyme complexes. So there's these huge complexes that work together to make the antibiotics. So we just take the idea is that we could potentially take bits from these different bacterial systems and put them together and make something completely new. But I think that's sort of the end goal of it, but it requires a lot of finding out the structure, finding out what these different bits do, and so we know exactly what we're going to be making. So my project was just to find that structure, and then once you know the structure, you can kind of hypothesize what that enzyme does, and then we know that we can incorporate it into new sort of pipelines downstream.
Amazing.
But yeah, that was very exciting. So I think I had two very interesting sort of research opportunities at the time, which I didn't even intend to do a PhD until my final year of undergrad, until I realized, because I enjoyed my sort of opportunity of working in that pharmaceutical company, and I knew that to go into that kind of field of research, I would need a PhD, and I just wanted to sort of improve my get more opportunities in research and see what else is out there. And I think I'm a very indecisive person, so I couldn't decide really what I wanted to do my PhD on. So, and I discovered this. So one of the PhD students who was supervising me under my undergrad studies, she was a Wellcome Trust student.
Okay.
And so I came across this program, the Wellcome Trust PhD programs. I'm not sure if you're aware of them.
Do you want to just give us a quick description?
Yeah. So they're essentially these four-year PhD programs, which follow sort of 3+1 method. And in the first year, it's usually you do different rotations. So that's typically 3 rotations in 3 different labs. And they sort of fall under one umbrella of themes or topics. And you can try out, like I said, and you spend 3 months in each lab, and you really get to know the lab, get to know the project, the research that you'll be doing, get to learn different techniques, and then you choose one of those which you prefer the most to stay in and do your 3-year PhD afterwards. So,
Does that, with that 3+1, does that integrate? Because I know some programs will integrate a master's degree.
So it depends on the university. At King's College London, it does. So the first year counts as a master's degree. So after each rotation, we had to write up reports and do presentations, and this all counted towards a master's degree, which is another plus, I think, of the program, because I think there's quite a big difference as well, because in a lot of other countries, you need the kind of pathway you go is undergrad, master's, PhD.
Yeah.
But in the UK, because many universities offer this placement year, universities regard that as enough or even more experience than you'd get in a master's degree. So you can go straight from having just an undergrad degree into a PhD program like this, which I think is something quite interesting that the UK does. And
Excellent. So then you've finished your biochemistry degree at Leeds, and we're coming into the PhD now. So so you said that you had this supervisor who was a doctoral student.
So this was in Leeds. She just introduced me to this Wellcome program, and so I looked at the different programs that the Wellcome had, and the one I was most interested in was this cell therapies and regenerative medicine program at King's College London, because I looked at the different projects that are available, and they were really all looking at sort of stem cell therapies, these new sort of regenerative medicine where we can use stem cells to kind of replenish damaged organs, or even create sort of new organs. And I think that there was quite a lot of interest in that field, but I didn't have much experience in that particular field. So I thought that doing the 3 rotations would be great to sort of get a flavor for what these are like. So I can give you a sort of brief description of my 3 projects and then go into the one that I chose for my PhD.
Yeah, absolutely.
So the first one that I did was I was actually using something called bioinformatics, which is where we sort of coding in a way, and you look at I was looking mostly at gene expression. So there's different ways that you can look at sort of the gene expression on the whole. You can do these sort of DNA microarrays, which is how it was done previously, but now we're moving more into something called RNA sequencing, which is where we sequence all the genes that are present in that sample, and you can do this in a bulk population of cells. So you just take a bunch of cells and you get the average expression for each gene in those cells, or you can do single cell, which is kind of new and more sophisticated technique. And you look at it individually within each cell, what the gene expression pattern is like, and you kind of evaluate that using these different software coding programs. So I was doing that and looking at how different genes can influence how susceptible a person is to HIV infectivity. So we were looking at, so we have this database, which is a collaboration between King's College London and the MRC and the Sanger Institute and a few other institutes, called the HIPC database. And it's essentially a database which has a lot of information on gene expression and the different phenotypes, which is how the cells look like, of these iPSCs, which are induced pluripotent stem cells. Mm-hmm. So we take skin cells, usually from people, from donors, and you convert them into stem cells. And that's this whole new inducing them to act as stem cells. So I was using this database that had their gene expression. So I was looking at how, in like a population which is quite diverse of different, these are, some of them were patients, some of them were healthy donor derived.
Sure, yeah.
And looking at how HIV can infect these cells. And I was looking at the genes that can kind of correlate with these outcomes.
So the idea then is, so say, I could turn up in your clinic or whatever. You could take some, I hope, healthy skin from me. Yeah. You can then somehow turn my fully differentiated kind of end of the line skin cell back into a pluripotent, but not totipotent.
No, it's a pluripotent, yeah.
Yeah, pluripotent stem cell, which you can then bathe in chemicals or do whatever you want to then turn it into a different type of cell. Is that the idea?
Yeah, exactly. So you could turn it into whatever kind of cell you'd imagine you'd want to turn it into, which is not, it sounds simple. It's not a very simple process.
It doesn't sound simple.
No, and usually it takes a lot of tries to try and get it to exactly the cell type that you want, and it takes a lot of time as well. But I was looking, so the lab, I was doing more of the bioinformatics side. So I had the data of how these HIV infects these different stem cells. So I was just analyzing the data using bioinformatics. That was my project. So then, which I really enjoyed. And now in my PhD, I'm also using a lot of bioinformatics myself.
Sure.
Too. Yeah. So my second project then was I was in between 2 different labs, and we were looking at, so one of them works with 3-dimensional gut organoids, which were quite cool.
Okay, we've got some new words flying around here.
Yeah. So organoids are essentially, like, they are mini organs which come from a stem cell. So in the gut, you have this sort of population of different cells, and within the crypts in the intestine, you have these stem cells at the bottom of the crypt, and they differentiate as they go along the crypts to the different kinds of epithelial cells you can get.
Sure.
So what happens is, if you can sort of isolate these crypts, so take them out, usually we were using mouse models, but you can also use those iPSCs that I was talking about and make human organs. And we put them in these dishes, and we grow them in this gel so that they really grow in 3 dimensions. So you see these kind of flowery 3-dimensional structures, which are sort of mini guts, which I think was quite interesting. And I was looking at trying to establish a in vitro model, so a model in a dish of the gut-brain axis. So I was culturing these organoids with sensory neurons, and just trying to establish how they communicate. And I think there's quite a lot of new research coming out now with how there's a lot of connections between the brain and the gut. And
yeah, absolutely.
It's highly innervated, and there's so many chemicals going up there. And because it's such an exposed organ to everything, to the outside world. I think it's a whole new area of science that I think is fascinating.
Incredible. So that was, so you were growing these sort of small human type digestive organs, as it were, in a dish, in a box, you know, and is the idea that they is it purely investigative? Is it to study, or can we, are we potentially going to be able to grow replacement bits of bowel for humans or for animals? What's the goal?
The idea for growing these organoids in the first place is that you can use them to replace pieces of the gut. So a lot of labs work with these gut organoids where they're looking at inflammatory bowel disease or Crohn's disease, and they're trying to look at how you can replace this damaged and inflamed gut tissue. And I think it is possible in some, so the Cleaver's lab, they work a lot with it's Hans Cleaver's, he developed these organoids, and they've shown that, they can be used to repopulate damaged tissues. So it's still, I guess, in the early phases, but for my project at the time, it was more exploratory, and I was just using them as a model to see how they can communicate with humans.
Amazing. So that was project number 2.
That was project number 2. And then project number 3 was the one which I've stayed on to do my PhD in.
Okay.
And I was looking at, so the lab, it works on CAR T-cell immunotherapy.
Okay, right.
Which is a very hot topic, I think, and you've probably heard something about it.
Yes. So just for reference, before you go, so Maria, the PhD student at Birmingham, research diaries that I interviewed before, I was given basically one instruction about this interview, and that was to delve into these CAR T cells. I should explore them. So please, as someone who, I only have the knowledge of T cells, that I have a molecular biology undergrad and I've done 3 years of medical school. None of this particularly means anything to me. So CAR T cells.
Yeah. So CAR T cells, they're kind of, I guess, they're a new kind of form of immunotherapy. I mean, so immunotherapy is this whole field of, we're trying to boost the patient's immune system to try and find the cancers and eradicate them. Because one of the issues with cancer is that cancer cells are very smart, and they can find ways to evade the immune system detection. So they can sort of hide in their own, like a tumor microenvironment, which is highly immunosuppressive. So even if there are immune cells there, they're kind of converted by the cancer to prevent any inflammation, to basically say, we're hidden, don't tell the body that we're foreign.
So they can't kind of mount an immune response, even if they find the tumor successfully, they can't actually do anything. Exactly.
They're kind of inhibited by these different molecules that these cells secrete, and they can't. So essentially, our bodies should be able to detect cancers. They'll see that cancers are foreign, and they should be able to remove them efficiently. But because cancers are smart in the way that they evade this, this is why they're allowed to persist, and this is why we see that they metastasize to different parts of the body, and they're kind of protected. They're sort of in this immune privileged zones. So then the idea of immunotherapy comes into sort of, we know what these cancer cells express on their surface, and we know to a certain extent. We're trying to understand what they express, and then we essentially design, you might have heard of monoclonal antibodies. So they're antibodies that then go and detect the sort of surface antigen, and then we essentially tell the immune cells, this is it, this is the cancer cell.
Again, just boys and girls at home, what do you mean by an antigen?
An antigen is this sort of molecule on the surface of the cell that's specific to that cell. So the idea with cancer immunotherapy is you want to find these molecules that are only expressed in the cancer cells but not expressed in healthy cells, because we want to train the immune system to know that this is what we want to eliminate, and we don't want to touch that healthy tissue. So it's just a molecule on the surface.
So I think that's again, for those at home, the common complaint with a lot of current cancer therapies, particular chemotherapies, is that they are, I think it's fair to say, indistinct in exactly what they will. You sort of point these drugs, which are very toxic, very damaging to tissues in the body, you point them at an area and they just indiscriminately kind of
Destroy.
scorched earth protocol, destroy everything,
Yeah.
which obviously for the body and for the people who have the treatment, not that good. So
Yeah.
there's huge advantage in these very specific targeted therapies. So you're looking to train the immune system to detect these very specific surface markers that only exist on solid tumor?
Well, I specifically work on solid tumors, but in general, the field is looking at any cancerous
Can be
malignant
anything.
cell.
So
Yeah.
what about
So
these
now
special T cells then?
our T cells, yeah. So T cells are kind of they're this immune cell, and you have different types of T cells, but the two kind of, I guess, main types you could call them are the CD4 T cells and CD8s.
Mm-hmm.
And if you read a sort of typical immunology textbook, it will tell you CD4s are the helper cells, and they will kind of help and guide the immune system, and CD8s are the killer cells, and they kind of, if they see something that's not right, they will just eliminate that cell. And that is to a certain extent true. And but as I said, these T cells, particularly the CD8s, are predominantly responsible for eliminating any cancerous or virally infected cells. So that's why we want to equip these T cells with a new receptor. Now, a receptor is a protein, which is again on the surface of a T cell, and it can essentially it can bind one of those antigens. So if we have a cancer cell which expresses a cancer-specific antigen, and we have a T cell we make it express a receptor that can specifically target that antigen, the T cell will go to the cancer cell, find that antigen. That receptor, once it binds its antigen, it transduces this activating signal into the T cell, and it says, you need to activate, you need to kill this cell.
Okay.
So we're essentially equipping T cells with like a weapon that they can go in, find the cancer, and kill the cancer cell.
So, okay, this raises some questions for me then. So, presumably in that case, because your cancer is expressing this particular surface marker, you need to tell the T cell what that marker looks like and give it, a key to the lock, or whatever the analogy would be.
Yeah.
Do you already have to know in that case what that antigen looks like in order for you to do that?
Yeah, so we would have to know what the antigen looks like, because the way that we design the receptor, and the way that usually CAR actually stands for chimeric antigen receptor. So, and chimeric means something analogous to what I was talking about previously with the bacterial systems. It's mixing, matching bits together.
Mm-hmm.
So it's a chimera, so we mix different parts of different receptors and we put it together. And the thing that targets the antigen or that binds it is usually an STIV, which is a single chain variable fragment, and it is sort of a part of this. Well, I guess you would call it. Yeah, it's a fragment that can specifically recognize that antigen, and so we put that on the top of the CAR so that it's only specific to that antigen. So once it binds it, it can relay signals through the other components of that CAR, which will then activate the T cell. So with a CAR, you typically have that fragment on top, which can find the antigen. Then you have some kind of bit that goes through the membrane of the cell.
Yeah.
Then in the inside of the cell, you have these, in the first generation CAR designs, you just had this activating component at the bottom. So it binds and activates. But then they found that because T cells themselves, they have these activating components, but they also have these co-stimulatory, which essentially boost the activity of, the activation of the T cells. So now the second generation CARs, they put in a few of these other co-stimulatory components. So ones that are typically used in CAR design are CD28 and 4-1BB, and they're essentially, we boost the activation of T cells by giving them the stimulatory aspect and the co-stimulatory components.
Okay, I may have misunderstood something then. So from the phraseology that you're using, when you say CAR T cell design, do these T cells, CAR T cells as their own entity, do they exist naturally in the body?
No, they don't. So I think I should have explained that from the start.
No, I'm just trying to follow. So they're just normal T cells that you would take. They're normal T cells.
Yeah. So the way that this works, and so now CAR T therapy has been in the clinic, and the way that it works in the clinic is you isolate T cells from patients, you genetically modify them and you make them express this synthetic chimeric receptor.
I see.
And
Right.
yeah, and then that's when they become CAR T cells, and then you infuse them back into the patient, and then hopefully those CAR T cells can then go and find the antigen.
So this chimeric region, this is a synthetic construct that you will design, in whatever gene structuring program you want to do, and then you sort of stick it. I see, and then you can culture a population of them, I assume, and then sort of.
Exactly. Yeah.
Okay, so what my next question is then, so let's say we have we've got a patient with some sort of cancer. Let's say they've got a really difficult, inoperable lung tumor, say, that surgery is a no-go. They can't get to it. Radiotherapy probably going to do more damage than good. So we're looking at immunotherapy. So our patient, what's the process? What has to happen to them?
To qualify for this kind of therapy, or
to take them through the process. What are they likely to experience if they have this treatment?
Okay, so it depends again on the type of immunotherapy, but if we're talking about CAR T cells in particular, then as I said, they would be so currently the two approved CAR T products, they're called Kymriah and Yescarta. They're for the B cell malignancies, so blood cancers.
Okay, sure.
There's none for solid tumors, which I can talk about in a second as well, why they work better on blood cancers than solid tumors.
Okay,
but
cool.
In the context of this scenario, you would they'd probably enroll on a clinical trial if there is one. And it would be the process of, as I mentioned, getting extracting the T cells from the patient, and then similarly to how we do in the lab, we would genetically modify these. So how we do it usually is we use viruses, but these are viruses that are not dangerous. So they're just we use them to be able to infect the T cell and make it, give it the genes that we want it to make, and that's what makes the receptor. And then once the T cells express this receptor, we can infuse it back into the patient, usually IV. So now if it's something like, if you want to target a solid tumor, you could also, instead of infusing it IV, you could inject it intratumorally. That would be, for example, that wouldn't be an option for a lung cancer, but potentially for like head and neck cancer or melanoma or something. So that's another way that you can ensure the CAR T cells get to the solid tumor. Now for lung cancer, I think it's still quite difficult to get the T cells to get to these solid tumors. And actually a sort of side effect of infusing T cells IV is that they can quickly get to the lung. So I think that's just another different effect because the lung is highly vascularized.
Sure,
so one of the side effects is that they can collect in the lung. But yeah, I think it's just, for now, for solid tumors, it's quite difficult to anticipate or to get these CAR T therapies to work, which is partly what my PhD project is on. Because, so if you imagine in blood cancers, these cells are kind of within the circulatory system, they're out in the open, and they express these antigens. And if we infuse the CAR T cells IV directly where the cancer cells are, they will immediately recognize and be able to kill them. Whereas in solid tumors, it's difficult for the cells first to get to the tumor. Then once they get there, it's difficult for them to navigate through that highly immunosuppressive, really hostile environment, the tumor microenvironment, to get there to eliminate the cells. But going back to your scenario, I think I'm sure there are clinical trials looking at the targeting of lung cancers using immunotherapies, but within CAR T cells, I think it's still being researched on how this can be done.
Amazing. Well, thank you. That was really, really interesting. I've learned more about T cells in the last couple of weeks than I ever wanted to know. But my next just few questions then are going to be a bit more practical, kind of about the ongoing situation and how it's affecting research. So how far through your PhD are you?
Okay, so I'm in my, I guess, first year of the PhD, but second year of the program. So
you're year 2 of 4 of the...
Yeah, of the 4. So I started in October 2019, officially the PhD. So still fairly new, I would say. But yes, so in terms of how this COVID-19 situation is affecting
Yeah,
the
of
research.
course. So if you're dependent on these cell cultures and seeing patients, and there's a lot
Yeah,
of interaction. Presumably, that's all a bit.
yeah. I mean, for my project, it's I'm not actually working directly with patients. I do most of my stuff in vitro, so in dishes within the lab. And we do some mouse work as well in the lab, which has all had to be stopped for now. So when something like this happens, I guess, I mean, I've only seen it happen once, thankfully, but we kind of just have to stop everything, freeze down all our cells and put all our animal experiments on hold. And I think it just depends on where people are at in their research. Some people are writing up their theses. In my case, I'm quite lucky that part of my project is this bioinformatics side. So I'm still looking at those gene expression levels that I was talking about in the beginning, more
Sure,
with RT rather than the HIV that I was looking at before. But
sure,
so that's what I'm trying to fill up my time with. But yeah, it's definitely going to have, I think, a large impact on research because a lot of our research depends on us. We have to do the lab work, and there's only so much you can do just from reading literature and doing these kinds of exploratory bioinformatical analyses.
sure. So I mean, from what I gather from talking to Maria, you'll have a thesis and potentially a viva due at the end of all of this.
Yeah, so you hand in a very lengthy thesis at the end, and yeah, the viva comes afterwards, which is essentially it can last anywhere from 2 to 4 or 5 hours of you sitting with two examiners, and them essentially questioning you. Reading, they would have read your thesis and they're going through the thesis and questioning you and asking you why you did things a certain way, and whether it can be done a different way, and these kinds of things to understand whether you really did your research, whether you understood the project, whether you uncovered something that's useful to the field, and I think just to see whether you deserve that title of having a PhD in the end.
And then, all going well, you come out the other side, Dr. Jana. What's what's the plan for you? Do you want to stay looking at T cells in the long haul? What do you think the future might hold?
Yeah, I'm not too sure yet. I think initially, so usually when you finish your PhD, you go on, if you stay in academia, you go on to do a postdoc in, I guess, in the same field. You can do it in the same lab or a different lab. But I think initially I would maybe do a postdoc, but I know this is a bit of a sidetrack now, but I'm actually looking at going into sort of, I guess, scientific venture or venture capitalism. So looking at funding or helping scientific and biotech startups. So there's
Mm,
so many people that work in these kind of new cell therapies and gene therapy fields, and people have all these ideas that I think they're really hard to communicate, and you can't really get them through to the big pharmaceutical companies, which will then help get it to FDA approval. But I think what I find quite interesting is now there's a lot of these sort of small biotech firms, and they usually start from a PhD student or a postdoc who has an idea, and they go and try and get some sort of funding for it and see and develop it into a new product. And I think that's an area that I'm really interested in, whether that will be pursuing my own company or working with venture companies that will be handling the funding of these other biotech
mm.
companies. I think that's something I'd be interested in doing because I think that's kind of where the future is headed in terms of healthcare and biotech.
I dread to ask. I mean, how I've read various figures on the cost of bringing things like this to market and getting FDA approval and all this. Do you have any, say your CAR T cell therapy, any estimate as to how much the total cost of that project, beginning the research to taking it as a licensed product to market?
I really couldn't tell you an exact figure. I know that they're in very high figures because if you consider for something like a CAR T therapy, it's not just the sort of preclinical research. So what we do in the lab, I mean, that itself costs a lot because all the techniques that we do, just in general, the things that we need for the lab are expensive. And it's not just the time, but I mean, it's not just the cost, but it's also the time that it takes to do all this research. But then once you come into something like clinical trial, you need to be producing these CAR T products in what we call GMP facilities, so good manufacturing practice. So it has to be
Hmm.
very clean, very sterile, because this is going into a patient. It can't be done in the same way that we do it in the lab. that's also very costly. Then it comes to keeping patients in the hospital and infusing these products and then keeping them longer in the hospital to monitor for any side effects. And that
Yeah.
all adds up a lot. And I think, it's quite easy to, in general, when you think of therapies or any kind of medicines, it's quite easy to say that big bad pharma makes everything very expensive and, they overcharge. But I think you really have to consider what the medicine is, because sometimes it's really, the costs are so immensely high that they do have to be priced high. So for CAR T therapies themselves are quite expensive, but the NHS is funding, I think, one of them that's
Okay.
FDA approved. Yeah.
Yeah. I mean, I read, and I don't know whether this is 100% true, but I think, for something like this, you're talking on the order eventually from start to finish hundreds of millions of dollars.
Oh yeah, probably.
Yeah, like I think the estimated, if any company were to try and take any drug from discovery through to manufacture and marketing, let alone an immunotherapy, you're looking at about $1 billion start to finish in the current climate.
In terms of immunotherapy, it's going to be even more expensive because this is not a therapy you can just go to a pharmacy and buy it from over the counter. It's something you'd have to be in a hospital setting for and get all this additional treatment and care that you don't usually need for a typical drug that you can get in a pharmacy. So I think there are some sort of limitations to immunotherapy, but I think it's quite an interesting and exciting field to think of this as, where we're using the patient's immune cells, we're just reprogramming them, reteaching them how to go and find and kill the cancer cells. So we're not really injecting any kind of toxic substances into the patients, and we can monitor the side effects. And I think it's
their own cells.
Their own cells, yeah.
Poetic. Amazing. Well, Jana, thank you very, very much. That is about where we'll wrap it, I think. It's been a really interesting discussion. I'm trying to get all the value for our viewers and listeners and people. And if you're thinking about applying to medicine, there's some really good stuff in there about how the research process works, why it's so expensive to bring new drugs to market, the whole set of processes that have to go on. I've learned a lot about immunotherapies that I didn't know. Not taught particularly in medical school. The details therein. It took me back to my undergrad, the days of biotech and stuff, and not handing things in on time. I think it's going to be
something that will hopefully get taught more in med schools, because I think it's becoming sort of the new standard of care compared to the traditional chemotherapy, radiotherapy for the treatment of cancer patients.
Amazing. Well, thank you very much. Are you happy for people to contact you if they have questions? Is that?
Yeah, sure. That's fine.
Okay, cool. I'll discuss with you, and we can put something out to go along with the video and the podcast that this will go out as well. So thanks very much, Jana. Take care, and I will hopefully speak to you another time.
Okay, thank you very much.
Take care. Bye.
Bye.
Thanks for watching, guys. There are 3 ways you can support the channel. The first one is to like, comment, subscribe, share this video with a friend. Just enjoy it generally. Second, you can buy me a coffee if you found it useful using my Ko-Fi link, which will help keep me awake during the editing process. And then thirdly, you can use my referral link to save 10% off your first year of Complete Anatomy 2020, my favourite 3D anatomy learning tool. Take care, guys, and I'll see you next time.